Hello viewers, today I will talk about the structure of prokaryotic cell. This is the general structure of prokaryotic cell. The living organism is divided into two main classes. Number 1 prokaryote and number 2 eukaryote. The prokaryotes are divided as archaebacteria and eubacteria. Eubacteria is the general bacteria. Today I will talk about general bacterial structure. This is the structural insight of prokaryote or general bacteria. General structure of prokaryote or bacteria include capsule, cell wall, plasma membrane, cytoplasm, ribosomes, plasmid, pili as well as flagella. Along with that, bacterial nucleoid is there. When we are talking about the bacterial cell structure, it includes the biochemical characteristics, morphological characteristics as well as structural overview. Let's first discuss about biochemical characteristics. The biochemical characteristics means the macromolecular contents of prokaryotic cell. The macromolecules include proteins, polysaccharides, phospholipid as well as nucleic acids. Proteins are found in flagella, pili, cell walls, cytoplasmic membrane, ribosomes as well as inside the cytoplasm. Polysaccharides are found in capsules, inclusions and cell walls. Phospholipids make the cellular membrane. Nucleic acids are of mainly two types, DNA and RNA. DNA may be the chromosomal as well as plasmid. There are three different types of RNA, mRNA, tRNA as well as rRNA. Morphological overview. Morphological overview includes the size as well as shape of the bacteria. Let's first talk about the shape of the bacteria. Bacteria may be either spherical or rod-shaped. Sometimes it may have some spiral or helicoidal structure. Let's first talk about the spherical structure, that is called as cocci. If the single coccus is there, that is called monococci. This is the structure of monococci. If two coccus join together, they are called diplococci. If the cocci are joined in a particular axis as a chain, they are called as streptococci, like this, streptococci. If they are arranged as grape-like structure, they are called as staphylococci. If four are together, they are called as tetrad. This is the structure of tetrad. If 8 together join as a cube, they are called as sarcina structure. So, this picture shows different types of cocci. This is the structure of sarcina under microscope. Let's talk about the arrangement of rod-shaped bacillus. Again, if single bacillus is there, that is called monobacilli. If two bacilli are joined together, they are called diplobacilli. If like this, they forms a straight chain, they called streptobacilli. But sometimes some ambiguous structure is there, that is called coccobacilli. They are somehow, the short rods are there, they are looking like the coccus. So, they are called as coccobacilli. Apart from this, sometimes the palisade-like bacillus are also there. It shows the types of bacilli. This is the picture of Bacillus anthracis and this is the picture of Bacillus subtilis. Spiral or helicoidal bacteria. So, this is the structure of spiral or helicoidal shaped bacteria. It may be either rigid or may be flexible. The rigidity may confer in the total structure or some part may be coiled. If total structure is rigid, in case of spirillum, it is total rigid structure. In case of Vibrio cholerae, some portion of the structure is coiled and it is actually the comma shaped. But in case of spirochetes, the total flexibility is found. In this picture, we can find the different spiral or helicoidal bacteria. Apart from all these general shapes, there are other two shapes also, stalked as well as budded shaped. Size. The size of the bacteria ranges from 0.1 to 100 nanometer. The smallest one is the Mycoplasma species. This is the picture of the Mycoplasma, having 100 to 200 nanometer in length. E. coli having the average size ranging from 1.1 to 1.5 micrometer. The largest one having 0.7 millimeter in length. The name of the largest bacteria is Epulopiscium fischelsoni. This is the structure. Now let's talk about the structural overview, which includes the external structure, cell surface, internal structure as well as some of the specialized cell structure. External structure. The external structure includes flagella, pili or fimbriae, capsule or glycocalyx structure. So external structure means the cell exterior. The first one is bacterial flagella. Bacterial flagella is needed for the bacterial motility. It is made by the contractile protein flagellin. It makes a hollow core tube. It is protruded from the cell surface and it has 90-degree bend as well. It gains its energy from the ionic flow and it helps in the motility of the bacteria. According to the different position of the flagella, there are different types. Number one is the monotrichous, if one flagella is protruded from one end. Lophotrichous, if a bunch of flagella is protruded from a particular end. The amphitrichous, the bunch of the flagella is protruded from the two ends of the bacteria. And in peritrichous, the flagella actually surrounded all over the surface of the bacteria. So, flagella is needed very much for bacterial motility. Bacterial pili. Pili is not yet needed for the bacterial motility, but pili having very shorter structure, it helps in bacterial conjugation, that is transfer of genetic material from donor cell to the recipient cell. So, this is the picture showing the function of pili. Pili is made by the protein pilin and it is very shorter structure than that of the flagella and it does not bend or weeps like flagella. Fimbriae. Fimbriae are having larger number than that of the pili and it surrounds all over the bacterial cell surface. Pili as well as fimbriae, both of them are needed to tether the bacteria over a particular surface like rock, teeth, etc. Fimbriae is made by the fimbrine protein and pili, fimbriae, all of the structure helps the bacteria to escape from the phagocytosis by WBC cell. So, in this way they can cause the disease or harm to the animal or other systems. Bacterial glycocalyx. This is made by the amorphous polysaccharide and it surrounds all over the cell surface. If the structure is larger, it is called glycocalyx. If the structure is smaller and well formed, that is called capsule. If it is not yet well formed, that is called slime layer. So, this is the picture of glycocalyx and this is the picture of capsule structure. Now, capsule as well as glycocalyx, everything is needed for the anchoring of the bacteria over a particular surface and again they help to evade, to escape from the immunological purposes and they actually helps the bacteria to avoid desiccation and during the loss of nutrient they actually helps the bacteria to overcome the situation. Cell surface. Now bacterial cell surface consists of cell wall, cell membrane, periplasmic space containing periplasmic flagella. Bacterial cell wall. This is the overview of bacterial cell wall. Let's talk about the components of the bacterial cell wall. The general feature includes the peptidoglycan layer. The peptidoglycan layer is made by N-acetyl glucosamine and N-acetyl muramic acid cross-linked with each other. In case of gram-positive bacteria, the cross-link is made by a cross-bridge, but in case of gram-negative bacteria, there is no such cross-bridge, only the interpeptide chain is there. The gram character, that is gram staining character, according to the different cell wall component makes the differentiation in between gram positive as well as gram negative bacteria. Apart from the general peptidoglycan structure, another ribitol derivative is there, that is teichoic acid in case of the gram positive bacteria, that is absent in case of gram negative bacteria. In case of gram-negative bacteria, we can find another outer membrane made by lipopolysaccharide, which is again absent in case of gram-positive bacteria. The lipopolysaccharide is made by lipid as well as polysaccharide. The lipid is called lipid A and the polysaccharide is called O antigen. That particular O antigen is the disease-causing element. And this lipid A is called endotoxin. When bacteria dies, then this endotoxin is getting released and causes the disease-causing symptoms like fever, shivering, etc. In case of gram-negative bacteria, within this outer membrane and inner cytoplasmic membrane, a particular space is there, is called as periplasmic space. This is the differential character of gram-negative as well as gram-positive cell wall. This is the gram stain picture of gram-positive as well as gram-negative bacteria. The first one is Staphylococcus aureus and the second one is Streptococcus aeruginosa. Bacterial cell membrane. This is the structure of bacterial cell membrane. Bacterial cell membrane has three components. Number 1 protein, number 2 lipid and number 3 is carbohydrate. The protein makes the channel, receptor, transporter and proteins are enzymes in nature. The lipid makes the fluidity of the membrane and the carbohydrate evoke the antigenicity of bacteria. It acts as bacterial cell surface marker. It can help to interact, to adhere with other types of cells. Periplasmic space. So, periplasmic space is the space in between the outer membrane as well as inner membrane in case of gram-negative bacteria, but sometimes may also find in case of gram-positive bacteria. This periplasmic space acts as reservoir of some of the enzyme in case of gram-negative bacteria. In case of some spirochetes, a particular flagella is protruded from the inside towards the outer membrane. They show some corkscrew whip-like motion and sometimes the outer membrane being flexible, this sort of flagella is helpful for their whip-like movement. This is the structure of periplasmic flagella. Internal structure. So, mainly bacterial internal structure is made by the cytosol. Cytosol contains ribosome, the genetic material as well as vacuoles along with some sort of inclusions are there. So, this is the general structure of the cytosol containing all the inner material. Bacterial ribosome. According to the centrifugation unit, it is 70S. It is made mainly by the ribosomal rRNA. 2/3 of the bacterial ribosome is ribosomal rRNA, but 1/3 is made by some of the protein. Bacterial ribosome is needed for the protein translation from mRNA. Bacterial chromosome is covalently closed circular DNA. It is called as nuclear body. Where we can find the presence of nuclear body within the cell or within the cytosol is called nucleoid material or nucleoid. We have to remember the bacterial nuclear material is not yet surrounded by any sort of membrane. That's why it is called prokaryotes, and that distinguishes itself from a eukaryote nucleus which has particular membrane which is absent here in case of prokaryote. So, this is the picture of nuclear material, nucleoid in case of prokaryote bacteria. Apart from chromosomal DNA, here we can find another extra chromosomal DNA that is called plasmid. Plasmid contains some of the luxury genes. Like in case of some bacteria, it contains the F factor which is needed for the fertility, for the generation of pili needed for the sexual conjugation, for transfer of genetic material from donor cell to the recipient cell. In another case, we can also find R plasmid. R plasmid contains the resistance factor against some of the antibiotic drugs. In case of E. coli, we also find col plasmid. Col plasmid gives rise to colicin, which can kill the neighboring bacteria. So, these are the maps for F plasmid as well as R plasmid. Let's talk about some inclusions. Mesosome is such an inclusion coming from the cytosolic membrane which acts like the cristae as mitochondria in eukaryotic cell. They act as the platform for DNA anchoring or binding during segregation. They can help to generate ATP by oxidative phosphorylation. Thylakoid. It is mainly found in case of some cyanobacteria. It is a strap-like inclusions and it acts as the surface for photophosphorylation. Gas vacuoles. Some thin protein membrane bound organelle. So, it helps to have some sort of gaseous material. It is found in case of some aquatic bacteria to maintain the buoyancy level. Carboxysome, again another vacuole-like thing which helps to fix carbon dioxide and to convert them to the organic matter. Poly-beta hydroxybutyrate granules, they act as the storage forms of some of the nutrient like glycogen as well as lipid and some of the organic butyrate material which helps in case of further nutrition during the depletion of the nutrition of bacterial or prokaryotic cell. Some storage granules are there called metachromatic granules which acts as the phosphate reservoir which is needed during the course of ATP synthesis. If the cell is stained with methylene blue, the metachromatic granules are observed as pink in color. Magnetosome. Again we find another inclusion body which is made by the crystal magnetite and it is helped during the course of magnetotaxis of the bacteria. Sulfur granules made by the elemental sulfur. The electrons are passed from the H2S and this sulfur granules is again the reservoir of the elemental sulfur which is needed by the redox reaction of the prokaryotic cell. Apart from this, some specialized cell structure are there. During the course of adverse environment, the bacterial cell adopt such the specialized cell structure. It includes the endospore, heterocysts or akinetes, conidiospore and spheroplast and protoplast. Let's first start with the endospore structure. When a bacteria is forced towards the adverse environment like the change in pH as well as elevated temperature, some of the bacteria like bacillus attains the endospore structure. Endospore comes out from the cytosol. First from the cytosol the forespore is made as well as smaller prespore is made. Then forespore engulfs the prespore and makes the endospore structure which is surrounded by two layers of the membrane. The outer coat is called cortex and the inner coat is called spore coat. Spore coat is made by calcium and dipicolinic acid. After maturation of the endospore, it releases out from the sporangium, right? And they are called as exospore. Within the spore, the DNA is very condensed and when the harsh environment is getting removed, the spore is getting germinated. So, this is the structure of endospore under microscope and general structure of the endospore. The endospore staining is done with malachite green. Heterocysts and akinetes, actually they are found in case of some cyanobacteria. In some cyanobacteria, for anaerobic nitrogen fixation, the heterocyst is made, and this akinetes is closely related with the heterocyst, which is less metabolically active. Conidiospores, in some filamentous bacteria, these sort of structures, specialized structure mainly, are found, where some cocci are attached with some sort of filamentous structure. So, this is the structure of conidiospore. When the cell wall is getting removed from the gram-positive as well as gram-negative bacteria and they are put inside some isotonic solution, some specialized structure is made, which is needed for some research purposes. In case of gram-positive bacteria, it is called protoplast. In gram-negative bacteria, it is called spheroplast. If it is not put inside the isotonic solution, the cell will burst, because cell wall as well as cell membrane helps to maintain shape as well as osmotic balance from outside external matrix from the cytosolic matrix. I have discussed about the structural hierarchy of a particular prokaryotic or bacterial cell. All the structures or some of the special structure may be act as the target site for the antibacterial drug antibiotics. Let's talk about them. The first one are the cell wall inhibitor. Penicillin, cephalosporin, they are the cell wall inhibitor. They inhibit the cell wall biosynthesis. Polymyxin B is cell membrane biosynthesis inhibitor. Tetracycline, erythromycin, streptomycin, they are protein synthesis inhibitor. So, I talked about the protein synthesis from mRNA by the help of ribosome. So, actually all these antimicrobial drug or antibiotics inhibit anyhow the bacterial protein synthesis. Quinolones are the antibiotics which inhibit the nucleic acid biosynthesis in case of prokaryotes. In conclusion, I have discussed about the biomolecular content of the bacteria, which makes the building blocks of prokaryotic cell. That means the amino acids to make the protein structure, the sugars to make the carbohydrate structure, as well as the nucleotides to make DNA as well as RNA. I have talked about the morphological content including size and shape of the bacteria. I have talked about the structural component including the cell surface appendages, the cell surface and the cytosolic components including the inclusions. I have talked about the structural component which acts as the target site for the antibacterial drug called as antibiotics. Thank you.